endomembrane system, one of the most fascinating and vital systems inside of a cell. The endomembrane system is responsible for creating, modifying, and transporting proteins to different parts of the cell or outside of the cell. In this video, I'll be demonstrating how the system works using a model, this animal cell that I drew, that simulates the formation and pathway of different types of proteins. My name's Tanvi Ramesh, and I'll be your tour guide on how secretory proteins like insulin, lysosomal enzymes, misfolded proteins, and glycoproteins travel through the endomembrane system. First, let's take a look at the formation of insulin, a secretory hormone, and how it travels from its synthesis to secretion outside of the cell. We're first going to start our journey off in the nucleus, where the gene for insulin is transcribed into mRNA. And from here, our mRNA is going to exit the nucleus and head on over to the rough ER, which brings us to our second step. Over here, these orange dots called ribosomes are going to translate the mRNA into a precursor protein called pre-proinsulin. And as our pre-proinsulin is synthesized, the protein is going to enter the inside portion of the rough ER, which is called the ER lumen. And this is where it folds and it turns into proinsulin, which once again undergoes some further modifications. So once we're done with that, we're going to go into our step 3, which is processing in the Golgi apparatus. But before we get there, we have to have a way to transport our proinsulin to the Golgi apparatus. And that's where transport vesicles come in. Transport vesicles are going to come from smooth ER or transitional ER, and I do that on the bottom over here. And over here, that's where these vesicles are going to come and help us transport our proinsulin to the Golgi apparatus. So I'm just going to draw that over here as our vesicle transporting our insulin. And obviously, this is going to contain more proinsulins, but just for the sake of this, I'm just going to draw it as one singular dot or one singular thing that's being secreted outside of the hormone. So once it's here at the Golgi apparatus, the proinsulin is now going to be transformed and modified into actual insulin. And what's ultimately going to happen, once again assuming that we have multiple of these proinsulins being transported at a time, Golgi is essentially going to set and package our little now insulins into secretory vesicles for transport and secretion outside of the cell. So our secretory vesicles are going to come and butt off of the trans-Golgi network. And that's going to happen over here. And we are going to have our secretory vesicles. And that takes us on to step 4, which is exocytosis at the cell membrane. So once we have our secretory vesicle containing our insulin, we're going to travel along the cytoskeleton until the cell receives a signal. And this is when the secretory vesicles containing the insulin are going to move up to the plasma membrane, and they're going to fuse with the membrane and release insulin from outside of the skull through exocytosis. Now let's take a look at how a lysosomal enzyme, like a hydrolytic enzyme, is produced and transported to its final destination, the lysosome. Similar to insulin, the enzyme is synthesized in the rough ER, and over here, once it's synthesized, it's going to go to the Golgi, where it's going to receive a special tag to be sent to a lysosome rather than secreted out of the cell. So over here, it's specifically tagged with a mannose 6-phosphate tag. And this helps the transport vesicles transport the enzyme to the lysosome over here rather than being secreted. So from the Golgi along the trans-Golgi network, the enzyme is transported to the lysosome, where it can function as a hydrolytic enzyme to break down cellular waste products of the cell. Not all proteins are successfully folded. Some proteins, such as misfolded proteins, need to be destroyed to prevent damage to the cell. So, assuming that this is going to be a protein meant for secretion, it's going to start its normal process from the nucleus to the rough ER. But if it's misfolded during this process, which I'm just going to mark as a bright green dot, if the protein is misfolded, it's going to be identified by chaperone proteins. And instead of being transported to the Golgi, it's actually going to retain in the rough ER. And then it's going to eventually be transported to a structure called a protosome. And this is just a rough sketch of a protosome. And this is eventually when it'll be degraded into amino acids. Lastly, let's explore how glycoproteins, such as cell surface antigens, are formed and transported to the cell membrane. The glycoprotein is also synthesized in the rough ER, where it undergoes glycosylation. And this is essentially the attachment of sugar molecules. And from here, it's going to be transported to the Golgi apparatus, where the glycoprotein is going to go and undergo further sugar modifications until it's prepared for delivery to the cell membrane. So once the glycoprotein is packed into its vesicle, it's transported to the plasma membrane, where it's ultimately displayed on the surface of the cell, and this allows the cell to communicate with other cells, playing a crucial role in immune response.